Development of neuronal circuitry of the human prefrontal cortex during the early fetal period. Structural organization. The first cortical cells appear in the neocortical preplate or primordial plexiform layer, figure 2.1a. Soon thereafter, the formation of the cortical plate, CP, at 8 postconceptional week, figure 2.1b, marks the transition from embryonic to fetal period, and prefrontal neocortical onlaga is now composed of 3 architectonic zones: the marginal zone, MZ, the cortical plate, CP, and the presubplate, PSP. These zones contain 2 main classes of neurons: radially oriented postmigratory neurons within the CP cortical plate, and randomly oriented and early maturing neurons located above and below the CP cortical plate, that is, in the marginal zone, MZ, and the presubplate, PSP, respectively. Between 13 and 14 postconceptional weeks, the end of the early fetal period, figure 2.1c, the deep part of the hitherto densely packed CP cortical plate transforms into a wider lamina that appears pale in Nissl-stain preparations and merges with the presubplate, PSP, thus forming a new prominent and transient zone, the subplate zone, SP. This marks the onset of the typical mid-fetal lamination pattern. Importantly, the early fetal cerebral wall, telencephalic pallium, shows early regional differences in the thickness and appearance of marginal zone, cortical plate, and subplate zone, MZ, CP, and SP, across medial, lateral, rostrocaudal, and dorsal ventral extent. Dorsal fetal pallium, as a forerunner of neocortex, displays thicker and condensed CP cortical plate, narrow MZ marginal zone, and wider PSP presubplate. The CP cortical plate becomes thinner toward the interhemispheric fissure, where dorsal pallium continues into the medial pallium. The pallium of the cortical limbus, hem, is thin and convoluted with wide marginal zone MZ, thin and convoluted with CP cortical plate, and almost invisible PSP presubplate. The CP cortical plate is initially not developed in the hippocampal onlaga. This early regional specification probably reflects the activity of intrinsic patterning mechanisms, whereby patterning centers generate across the dorsal telencephalon graded expression of transcription factors acting on cortical progenitor cells. Thus, the frontal telencephalon becomes specified by specific transcription factors such as the basic fibroblast growth factor, lowercase b, capital FGF, expressed in the ventricular zone of the rostral anterior telencephalon. Although this early patterning of the cortical protomap occurs in humans probably during the 2nd month of embryonic life, that is, even before the formation of the cortical plate, the specification of cortical areas continues during the fetal period, and thalamic input seems to have a significant role in the final differentiation of cortical areas. Neurogenetic events. Major neurogenetic events are production of young postmitotic neurons in the ventricular zone and their migration through the intermediate zone, figure 2.6. Comparison with data in monkey shows that in early fetal period, neurons destined for superficial or associative layer 3 are not born yet. Molecular specification of early cortical neurons was proven using different markers for GABA and renin. For studying early cortical development in a clinical setting, it is important that early transient proliferative, migratory, and synaptic zones were successfully visualized by in vivo in utero imaging around 13 postconceptional week. Neuronal circuitry and functional organization. The neuronal circuitry of the early prefrontal cortex consists of a small number of synapses, dendrites of postmigratory neurons, and presynaptic axons arising from modulatory extrathalamic subcortical systems. As described for the midlateral telencephalic wall, and then the reference Molliver, Kostovich, and Vanderloos, 1973, and the anterior cingulate cortex, Kostovich and Krempotich, 1976. Of human fetuses, early synapses display bilaminar distribution and are located above and below the cortical plate. Such bilaminar distribution of early synapses was also demonstrated in equivalent stages, embryonic day 60, of fetal rhesus monkeys, in Bourgeois, Goldman, Rakesh, 1994. In the developing human cortex, prospective postsynaptic elements for the early synapses were revealed by Golgi impregnations. References for that are 1988, 1990, 1992, and electron microscopy, 1990. The most likely candidates being neurons of the subplate zone, 1974, 1990. However, radially oriented neurons of the immature cortical plate also extend their dendrites in the marginal zone and the pre-subplate, and thus represent putative postsynaptic sites for at least some synapses. There is substantial evidence for the presence of presynaptic axons within early synaptic strata of the human cerebral cortex. Modulatory afferents arrive early from the monoaminergic brainstem tegmentum and cholinergic basal forebrain. Although thalamocortical axons are still in the stage of pathway selection, their involvement in the early circuitry cannot be excluded. Short presynaptic input is also provided by subplate neurons that express glutamate and GABA, as well as several neuropeptides. Functional organization. At this early age, supragranular neurons involved in long cortical-cortical networking necessary for cognitive functions are not born yet. There are no imaging or functional recording studies of the early human prefrontal cortex. Therefore, we have to rely on experimental studies in animals with similar patterns of circuitry organization. These studies indicate that subplate cells communicate with both non-synaptic and synaptic contacts and produce spontaneous endogenous oscillations that do not depend on sensory input. These results suggest that long before the onset of cognition, the prefrontal cortex of early human fetuses has neuronal circuitry involved in spontaneous endogenous activity.